Our next presenter is Nicole Sanabria, geologist with Michigan's Eagle, presenting virtually.
All right, so I will be discussing a project conducted by the state of Michigan, which involved collecting soil samples to understand the presence and concentrations of PFAS in Michigan forested areas via composite sampling. I'm here representing Eagle. I'm a geologist for Eagle, and I'm also a graduate student for Western Michigan University, and also kind of put this report together as my thesis as well. So in 2019, the Michigan PFAS Action Response Team allocated funding to assess the presence of PFAS in various land use types and to test out the utilization of a modified incremental soil sampling method, specifically for collecting PFAS samples. The forested land use type was chosen to be sampled first due to ease of access, and as all of these samples were collected on state-owned land. The forested land use type was identified using the U.S. Geological Survey National Land Cover data from 2011 and was defined as at least 85% coniferous, deciduous, or mixed forest. All samples in the forested areas were collected as of the end of 2021, and it's important to note that our study was not a background study. So using the public land survey system, the state was divided into 40-acre parcels, and parcels were at least 85% represented. Cumulative coniferous, deciduous, or mixed forest comprised the population of the forested land use cover. And one sample was collected from each of Michigan's 83 counties. All forest samples were located on state-owned forested land, and they were in easily accessible areas. The roughly 240 sites of known PFAS contamination were not considered when selecting sampling locations. Soil samples were collected utilizing a modified incremental soil sampling method from a depth of 0 to 6 inches below ground surface using a 2-inch diameter coring tool. 9 aliquots were collected from 50 by 50 foot grid, which yielded 1,800 grams of soil. And sampling was conducted following the Michigan DEQ soil PFAS sampling guidance, which we established in 2018. So the chemical analysis of PFAS, metals, and TOC was conducted by Eurofins Test America Laboratories, and the geotechnical analysis was conducted by SME. For PFAS analysis, the aliquot size was doubled from 5 to 10 grams for the limit of quantitation to achieve lower detection limits. PFAS were analyzed using the modified EPA method 537 with isotope dilution, and samples were analyzed for 28 PFAS compounds and 26 metals. So the results of our study, at least one PFAS compound was detected in all forest samples except for one. Compounds detected most frequently are shown in the table at the right, with PFBA being the most frequently detected, followed by PFNA, PFOS, and PFHPA, and so on. Compounds or PFOS was generally detected at the highest concentrations, followed by PFBA and then PFOA. And total PFAS concentrations ranged from non-detect to 2.016 micrograms per kilogram. So this was the distribution of total PFAS concentrations in the lower peninsula. This like pale yellow dot up there in the northern part of the lower peninsula is the non-detect result. And then as you can see on this map, a lot of the higher detections were located in the southern half of the lower peninsula. And then here's the upper peninsula, generally lower concentrations than what we saw in the lower peninsula. So the sum of carboxylic acids was higher than the sum of sulfonic acids in 79% of the samples collected. There were no detections of fluorotelomer sulfonic acids, fluoroalkyl ether compounds, or perfluoroalkane sulfonamides in any of our samples. A statistical analysis was conducted as part of this project. Principal component analysis was primarily used to assess similarity in PFAS concentrations between samples in regions of the state. PFAS with greater than 75% non-detects were excluded from this analysis, leaving the 10 PFAS compounds listed here. Regression analysis was used to determine if the presence and amount of one variable, such as TOC, moisture, clay content, or a specific metal, was correlated to any single PFAS or total PFAS. Given the significant range of non-detects we had, our detection limits for the non-detects, which range from 0.11 to 37 micrograms per kilogram, we did create imputed values for the non-detects. So PCA was conducted using a correlation matrix of standardized data, and samples were given one of 3 regional designations to see how component scores varied across the state. The Lower Peninsula was divided into a North and South region, which is NLP and SLP respectively. The primary component of variation, or PC1, contributed 41% of the variance, with PFNA, PFHPA, PFOA, and PFOS having the highest loading factors. And the secondary component contributed 14.2% of the variance, with PFUNA, PFBA, PFBS, and PFHXA having the highest loading factors. As you can see on the score plot, samples from the UP and Northern Lower Peninsula seem to be clustered together, indicating some similarity between these samples, whereas samples from the Southern Lower Peninsula are quite scattered across the plot, so indicating more variance between those samples. For the regression analysis, 5 PFAS were assessed, including PFBA, PFNA, PFOS, PFHPA, and PFOA, which were our 5 highest detected compounds, as well as total PFAS concentrations. Concentrations of PFAS were compared to 18 metals, TOC, moisture, and clay content using a linear regression model to determine if any correlations existed between any of these factors. No significant correlation was found between PFAS and metals, TOC, moisture, or % clay. The strongest correlation existed between lead and total PFAS and zinc and PFBA. But even here, a review of the residual plots showed unequal variation, clusters, and large residuals, suggesting there may not be a strong, significant correlation there. Results of this project were compared to results observed in other soil studies that assessed PFAS concentrations either in a forested or similar land use type, and were not associated with some known contaminated site. Results from the Michigan project were generally consistent with the results of other studies, and in some cases were significantly lower. Results may differ due to the number of compounds analyzed, samples and sampling and analytical methods, and the location of the samples. Although many of these studies intended to collect samples away from known contaminated sites, our knowledge and understanding of the use of these compounds in industry has greatly evolved over the last few years. So it's possible that some results of these studies were influenced by nearby sources that may not have been known to use PFAS at the time of collection. Most of the higher PFAS concentrations observed during this project were located throughout the southern half of Michigan's Lower Peninsula, which is not really surprising given this is the most populated and heavily industrialized region of the state. Approximately 80% of Michigan's known PFAS contaminated sites are located in this area. PFCA concentrations were higher than PFSA concentrations, which was also observed in most other PFAS soil studies. Although no significant correlations were found between PFAS, TOC, moisture, and clay, in this project, that may be due to a lack of variety of soil types observed in samples. Sand and silty sand were the primary soil type identified in 75 out of 83 samples, with moisture content below 25% for all but 13 samples, and clay content below 10% in all but 11 samples. Considering PFAS were detected in almost all of the forest samples in the study, atmospheric transport appears to be influencing the presence of PFAS in soils, but it's currently unclear whether this is due to nearby PFAS sources or because these compounds are present in the atmosphere at a larger scale. Obviously, significant data gaps remain regarding PFAS fate and transport in soil and the atmosphere, so further research is needed in this area. This could include air and rainwater sampling, sampling at greater depths in soil rather than just at the surface, which is what most of the soil samples, how they have conducted their sampling, and understanding precursor transformation in the environment. Like I mentioned earlier, the results of this study are not supposed to represent background. Because samples in the study were only collected on state-owned forested lands, results may not be representative of all forested lands in Michigan. And again, most importantly, no consideration was given to known PFAS contaminated sites. And it's extremely likely that some of the results in the study were influenced by nearby sources. So in conclusion, this project demonstrated successful implementation of incremental soil sampling methodologies for statewide sampling of PFAS, and lower detection limits were achievable by increasing the aliquot size, making it possible to compare our results to the Michigan soil criteria and have a clear understanding of what concentrations are present in forested soils in our state. No significant correlations were observed by PFAS and TOC, moisture or clay content or metals. And again, atmospheric transport appears to be contributing to the presence of PFAS in soils in Michigan, although further research is needed to understand this. There is a report on all of this data. It's going to be available on the MPART site, but I'm not exactly sure when. So until it is, feel free to reach out to me if you would like a copy of the report or have any questions on this presentation.
Great, thank you.